This is the second part of the lecture on nucleic acids. We're picking up with part 2, the structure of nucleic acids. Remember that nucleic acids include the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. The monomers, or building blocks of nucleic acids, are called the nucleotides. Each nucleotide is made of 3 parts, a phosphate group, a pentose sugar, and an organic base called a nitrogenous or nitrogen base. Here you see the generalized structure of a nucleotide. This is the phosphate group. Phosphate groups include a phosphorus with 4 oxygens attached. One of the oxygens of the phosphate is attached to the 5' carbon of the pentose sugar. Here is carbon 1, 2, 3, 4. This is 5. And this is the oxygen in the ring of the carbon. This is one of the 2 kinds of nitrogen bases. This nitrogen base has a double ring. The green in here is nitrogen. And here is the other kind of nucleotide, again a phosphate attached to the 5' carbon of the pentose sugar, but the other kind of nitrogen base has only a single carbon ring. So our 2 types of nucleotides are named for those bases. The pyrimidines have the smaller single ring of carbon. These are cytosine and thymine in DNA. In RNA we find uracil instead of thymine. The larger 2 carbon ring nitrogen bases are the purines, guanine and adenine. Here you see the 4 nucleotides of DNA. Please notice in this diagram that there are 3 phosphate groups attached to each of them. As they are used in the production of DNA, 2 of the phosphates would be removed, and the additional energy released helps to build the molecules of DNA. So here we see the pentose sugar, which in the case of DNA is going to be deoxyribose. Notice there's not an oxygen on this carbon. And here we see our 4 nitrogen bases. These are the 2 purines, adenine and guanine. These are the 2 pyrimidines, thymine and cytosine. Small differences that you will not be tested on allow us to distinguish adenine from guanine. So if you notice here on this carbon there's an amino group attached, whereas on this carbon and guanine there's a double bonded oxygen instead, and the amino group is on the side. Those amino groups have an effect of being a base. They elevate the pH. So again, in the pyrimidines, only 1 carbon ring. In cytosine, there's an amino group, where in thymine, there's a double bonded oxygen. There's still an amino group here. So the structure of DNA is characterized as a double helix. That is, it has 2 strands that spiral around each other. The 2 strands have opposite polarity. The phosphates that attach one nucleotide to the next are going to attach from the 5' carbon of one pentose molecule to the 3' carbon of the next. And this provides the directionality of the 2 strands. So the 2 strands run in opposite directions. One strand has the 5' carbon at the top and the 3' at the bottom, and the other one is flipped, 3' to 5'. The 2 strands are held together with hydrogen bonds between the bases. Bases always bond the same way. Adenine bonds with thymine, held together with 2 hydrogen bonds, and guanine binds with cytosine, held together with 3 hydrogen bonds. We know the dimensions of the DNA molecule based on the X-ray diffraction that was pioneered by Rosalind Franklin. So we know that the entire double helix is 2 nanometers wide, and we know there are 3.4 nanometers between turns, and each turn of the helix contains 10 base pairs. So in this diagram we're seeing the directionality of the 2 strands, and also we're noting the numbers of hydrogen bonds, 3 between guanine and cytosine, 2 between adenine and thymine. So here you can see the pentose sugar, deoxyribose, with the carbons numbered. So this is the 3' end. The 5' carbon of this pentose sits right here attached to the phosphate group. That phosphate attaches to the 3' carbon of the next pentose molecule. Here's its 5' carbon, Here is the phosphate group that runs between that 5' carbon and the 3' carbon of the next pentose. So it's 3' at this end and 5' at this end. The opposite strand runs in the opposite direction with the 3' carbon here closest to the 5' carbon of the matching strand. So this is 3', here's your phosphate, 3' to 5'. Here we can see those measurements that were pioneered by Rosalind Franklin's X-ray diffraction technology. The entire width is 2 nanometers. 1 turn of the helix is 3.4 nanometers, and 1 turn contains 10 base pairs. So the distance between each base pair must be 0.34 nanometers. Again, you can see 3 hydrogen bonds between guanine and cytosine, and 2 hydrogen bonds between adenine and thymine. Watson and Crick sent their very famous letter to Nature in 1953 and couldn't resist the temptation to propose what was obvious to anyone who looked at the structure, which is that each DNA strand could act as a template or pattern for replicating its opposite strand. They had no evidence for this, but the implications, I think, were pretty apparent to everyone, and they wanted to make sure they got the credit for it. Now, not that much later than Watson and Crick were working, Arthur Kornberg was working with E. coli bacteria, and he was doing enzyme assays of DNA, and he extracted an enzyme that he demonstrated was necessary for making DNA molecules from the nucleotides. He called this DNA polymerase because it turned the nucleotides, the monomers, into the DNA strand, the polymer. Later, other enzymes were found, and this being the first one to be discovered was called DNA polymerase I. So what Kornberg did was he added a single DNA strand to a medium along with some of his enzyme that he isolated, the DNA polymerase I. And he also added some nucleotides that were radioactively labeled with carbon-14. So he allowed the experimental mix to work, and at the end of the experiment, what he found was that the DNA strand that he had started with was still there, but now it had a partner strand that was made entirely with the radioactive carbon-14. Kornberg won the Nobel Prize in 1959 for his work. Now, at about the same time that Kornberg was being recognized for the work that he did, Matthew Mieselson and Frank Stahl designed an elegant experiment that provided conclusive evidence that showed DNA replication was semi-conservative. The term semi-conservative means that each strand forms the template for the complementary strand, the matching strand that's made from new nucleotides. And if you'll look at this link, you can get to that link through the PDF copy online of the PowerPoint. It's definitely worth watching. So what Mieselson and Stahl did was that they grew bacteria exclusively on radioactive nitrogen as a source, and eventually got a colony of bacteria where all the nitrogen in the bacteria was radioactive or heavy nitrogen, including the nitrogen in the DNA molecules of these bacteria. Then they took bacteria out of the heavy nitrogen and grew them in light nitrogen or non-radioactive nitrogen, nitrogen-14. And in one generation, that is, after giving the bacteria time to replicate themselves just one time, they analyzed the DNA and they found that the DNA was half heavy nitrogen, radioactive, and half nitrogen-14, non-radioactive, light nitrogen. So they knew from this experiment that half of the original nucleotides were retained and the other half were taken from new nucleotides made from the light, non-radioactive nitrogen-14. So here you can see the diagram that shows what Mieselson and Stahl did. Watch the video, it's better. But here you can see that they started with radioactive double strands, grew them in nitrogen-14, and were able to demonstrate that exactly half, that is one strand of each of the new DNA molecules that were formed in the replicating bacteria were made of the light non-radioactive nitrogen-14. So Michelson and Stahl are widely praised for the elegance of their experiment. Remember when they were working, the 1950s. So here we see what semi-conservative DNA replication looks like, as we know. Before, you've got the two strands, C with G, A with T. During replication, the two strands have to separate, and as they separate, new nucleotides are brought in to match the existing ones. So what you wind up with when you're finished is two exactly identical DNA molecules. Each of them retains one of the old strands, and each has produced a new strand to match the other old strand. So we're going to go through the steps as well as we know them. I am going to provide a link, look for it in the module, to a really great animation of semi-conservative replication. The base pairs we know are complementary. Remember what that means. That means A always matches with T and G with C. They polymerize only in the 5' to 3' direction, and this is just a peculiarity of the enzymes that we have that will only allow replication in one direction. The first enzyme we need is called a helicase. Helicase is going to unwind the double helix, creating what we call a fork in the helical structure. And we're going to then take those single strands, we're going to add some binding proteins to stabilize that unwound molecule. And notice now we're going to use a different polymerase, DNA polymerase type 3, and it's going to add nucleotides only to what we call the leading strand, the 5' to 3' end. And these are added continuously because that's the right direction. We also have another enzyme called a primase. Primase is going to synthesize a little RNA primer on the lagging strand, the one that works backwards. And then DNA polymerase 1 can add nucleotides in little short segments to this backwards facing strand. These short sections are called Okazaki fragments, and they're named after the guy who first figured out how all this works. Well, that's all very confusing. Let's look at a picture. So here you see the DNA polymerase 3 can add continuously to the 5' to 3' strand, but this other strand that's going backwards has to have this combination of other enzymes. So here in this diagram you can see if we're adding them on 5' to 3', the leading strand, as this unzips, we just keep adding one nucleotide after another. But on the lagging strand, what we find is that we would want to add them in this direction, but they can only be added in the opposite direction. So what you wind up with is short segments, those Okazaki fragments, that are put together, and then we're going to use another enzyme called a ligase to seal the gaps between them. Very complicated, very clumsy, definitely a sign of natural selection at work. So ligase is going to seal our Okazaki fragments to each other. Remember that we get the energy from those phosphates that on the free nucleotides, they're like ATP, GTP, CTP, and TTP. And when you take the extra two phosphates off, it releases energy just like ATP does for chemical reactions. And that energy then can be used to add the nucleotides onto the growing strand. Well, you can imagine that it's easy to make mistakes. So we also have enzymes that proofread the DNA, and if they see a mistake, they'll fix it. If they don't fix it, then it is going to persist. And once it persists, it's going to get copied from then on. And those persistent changes are called mutations. A mutation typically is going to be something that causes a problem with the resulting DNA. Either it's going to cause the protein to be misshapen, or the protein not to be produced, or the gene never to be turned on. And so you wind up with a missing protein. If it's a vital protein, the cell will simply die, and the mutation will be gone with it. But some mutations, in fact, turn out to be advantageous, and those then can persist, and they are the basis of the changes in the genome that provide the raw material for natural selection to work. DNA is replicated during the S phase of the cell cycle. So recall what we've already studied with mitosis and the cell cycle, and remember that the S phase comes shortly before mitosis begins. So one of our DNA polymerases can repair problems that crop up during replication. And remember, we also have our inhibiting factors, our checkpoints in the cell cycle, that if there's a problem, we can stop cell division, we can proofread and repair the errors if they are caught. Mutations are typically produced by exposure to things like ionizing radiation, which could be ultraviolet radiation, chemicals, heat, even viral infections can cause mutations to DNA. And the mutations that are most likely to survive are the ones we call spot mutations, where they are simply substituting one base for another base. For example, cytosine can lose its amino group. Here you see cytosine, there's its amino group. And here's thymine, that instead of an amino group has a double bonded oxygen. So sometimes radiation, for example, could ionize, hit that little amino group, and cause it to be converted from cytosine to thymine. And then it will be mismatched, and if it doesn't get caught, it will persist. That's a spot mutation, and an example of that is a disorder we already studied, HYPP, in quarter horses. Remember, we studied the history of the champion stallion Impressive, whose semen was sold to increase his genetic contribution to the next generation, and many of those horses were found to be defective and had to be put down, all from one change from a cytosine to a thymine. Most mutations can be repaired unless they occur right when the cell is dividing. And we'll look when we get into genetics a little bit more about how those mutations can occur. But we do have as many as 50 different enzymes that have been identified as having the ability to locate and correct errors. Okay, so we have a handle on DNA in the nucleus of the cell, but don't forget that at least 2 organelles also have DNA. This contributes to the current theory of endosymbiosis, which, again, this link will be found in the PDF of the PowerPoint that's posted online. This theory is in a state of change, so I'm going to be addressing that in class when I come back next week. We do find DNA in both mitochondria and in chloroplasts. We haven't really looked at chloroplasts much this semester, so we'll worry about that later. But we know that the DNA of both mitochondria and chloroplasts is very similar to the DNA in prokaryotes. And in fact, we've already looked at that this semester. This is an image showing actually the genes found on mitochondrial DNA. It's very short, but it is a circular segment, and it is attached to the internal membrane of the mitochondria, just the same way bacterial DNA is circular and attached to the inner membrane. So the fact that mitochondria and chloroplasts have their own DNA allows them to replicate. It not only allows them to replicate, it requires that they replicate to get that genome to persist. So the question then arises, did the ancestors of these organelles live an independent life? And the answer so far appears to be yes. We've actually found genomic similarities between mitochondria and a specific group of free-living bacteria, and likewise between chloroplasts and the cyanobacteria that we know also perform photosynthesis. All organelle DNA is contributed by the mother because the sperm donates only nuclear DNA. Only the nucleus of a sperm is typically injected into the egg when an egg is fertilized. So all of the DNA in the organelles comes from the mother. All mitochondrial DNA is maternal DNA, and the same for chloroplasts in plants. So we use mitochondrial DNA in evolution studies and, interestingly, in forensics. So in 2012, this grave was dug up. It was announced in 2013 that they were pretty sure it was the grave of Richard III. You probably don't remember enough of your British history, but Richard III was a king of England at a very turbulent time. He was notable for his strength in battle, but also because he had a very crooked back. So when they uncovered bones in this parking lot, in the very spot where they knew from historical records that they suspected Richard III had been very hastily buried, it had been the site of a friary and was later turned into a parking lot. When they found skeletal remains of an individual with a crooked back who had died with injuries consistent with the historical records, a blow to the head, they took the DNA for analysis. Well, the problem is Richard III lived a long time ago, but records are kept, and so some of his maternal descendants were known. At least two of them were known, one of whom was willing to come forward and say, yeah, I'm a descendant of Richard III. So they took mitochondrial DNA because it was from a living female descendant of Richard III. So they would share mitochondrial genetics, and the match confirmed the identity. Apparently the mitochondrial DNA was unique enough that they could tell this mitochondrial DNA from any other. Even more interesting, they also took DNA samples from the Y chromosome. Again, they can figure that out based on the Human Genome Project. They know the genetic sequences of the chromosomes, and they found that it did not match, which means that someone who thought he was a father of the king or in the king's lineage somewhere was not the father. Very interesting. This concludes this rest of the lecture on DNA structure. Be sure to watch the lecture on protein structure, which I'll be posting next.